My name is Brad Zuchero. I grew up in the woods of Pennsylvania and then went to Vassar College up in Poughkeepsie, New York, because I wanted to double major in biology and music composition. But then there were way too many interesting things to study, so I didn't end up double majoring. I just stuck with biology and took lots of music and Eastern religion, all sorts of fun stuff. While I was there, I became really interested in research. I got the bug for doing research by spending a summer at Vassar. And then I applied to a bunch of summer programs for undergraduates and ended up getting a position at UCSF in San Francisco for one summer and then one summer after that. And that made me fall in love not just with research, but with California. So then for grad school, I applied only to schools on the West Coast, ended up coming back to UCSF for my PhD. And when I was doing my PhD, sort of the options were open about what I could work on. And I was initially dead set on working on one particular thing. This is a phenomenon called RNAi, which people probably still know about, but now everyone's talking about CRISPR. In 2001, RNAi was the most exciting thing, I think, in molecular biology. And so I wanted to work on that. But then I did one of my laboratory rotations, which is something that we do as first year PhD students. You try out a couple different laboratories to find your home. And I went to a lab where I wasn't necessarily excited about the topic, but I loved the lab culture and the mentorship the PI was sort of building in this lab. And so I ended up joining that lab. This was Dyke Mullen's lab at UCSF, and they studied actin biochemistry and cell biology with the overarching goal of understanding how cells in our bodies change shape, move, crawl around. I fell in love with that topic after learning more about it. And I'd say, this is my advice that I always give to young scientists is the mentorship and the environment that you're in is almost way more important, I would say, than precisely what you're studying, because at least for me, I'm a curious person. I could fall in love with any research topic once I know enough about it. And so I did my PhD trying to understand how cells change shape and move. I discovered a protein that regulates this in vertebrates. And then for my postdoc, as you know, for faculty, people who want to do faculty jobs, get
Yeah.
faculty jobs, right? PhD is not enough. You have to do more training. This is even true for a lot of biotech jobs. I was interested in neuroscience, but I wasn't 100% sure that I wanted to go into neuroscience. So I asked a bunch of people who to rotate with or who to look at for postdoc labs. And the name Ben Barres kept coming up. And Ben Barres, I didn't know about Ben Barres, but I started reading about Ben Barres and I realized that this is a really exciting direction to go in. So Ben was a professor at Stanford. He died a few years ago from pancreatic cancer. He was the person who put glial cells, the dark matter of the brain, on the map, but also was an amazing mentor and advocate for women and underrepresented minorities in science because he was transgendered. And so my decision to go into glia, which is what I do now, came really from just wanting to be mentored by Ben. And that was an amazing decision. And so here I am now, Stanford professor studying glia, specifically the myelin-forming glial cells of the nervous system. And the road to get here, is it's circuitous, and it was informed mostly by mentorship and wanting to find the right people to work with rather than the topic.
Do you mind explaining a little bit more detail what your research is about?
Glia comes from the Greek word for glue because traditionally neuroanatomists, going all the way back to Cajal, as they were characterizing the nervous system, they saw neurons, they put together the neurocentric dogma of how the brain works or the nervous system works by neurons firing, and then basically they ignored the glial cells and thought of them as just brain glue that holds everything together. What Ben is really famous for, Barris, my mentor, he and many others in the field, especially many of his trainees who he supported, kind of turned that neurocentric dogma on its head a little bit and found that glial cells are actually really important. They regulate the development, the dynamics, and diseases of the nervous system. When I say glia, what I'm talking about is all the cells in our nervous system that aren't neurons. There's a couple major types. There's microglia, which are the resident immune cells of the nervous system, astrocytes that play diverse roles in supporting neurons, but also actively controlling the formation and elimination of synapses. And then there's the oligodendrocyte cells, and those are the cells that my lab primarily focuses on. Oligodendrocytes build the myelin sheaths in our brains and our spinal cords throughout the central nervous system. Myelin, sort of like glia, was long thought to be kind of a passive thing. It's just something that's laid down during development. It's important, but it's just boring insulation, right? I have my wire here. The wire insulation on this wire, not particularly interesting. But it turns out that this insulation, this myelin that is built by oligodendrocytes, is important for the development, but it's also actively supporting neurons, and it's incredibly dynamic, and it can change its physical properties in response to neuronal activity or in the context of learning. The emerging idea is that these changes in myelin are just as important as changes in synapses that occur during learning. I always like to say, if you take a step back, the nervous system, a circuit of neurons connected to each other, there's many different knobs that you can tune to change the dynamics of the system. One is you can change the strength, or position of the connections, the synapses. But another thing you can do is change the speed at which the information travels through the axon. And that's what myelin and myelin dynamics are able to do. This is not in textbooks yet because this is an emerging, exciting idea. Michelle Mange, who's a professor here at Stanford, she coined the term adaptive myelination as this concept that myelin is actually able to change. And so there's a couple ways that this can happen. One, turns out throughout our central nervous systems, you more than me actually have a lot of unmyelinated stretches of neurons that have the capacity to get myelinated. And so
Yeah.
the addition of new myelin that allows for or promotes or supports new learning. Another form of myelin dynamics is that existing myelin can change its properties. So the thickness of the wrapping, if you can sort of picture this is not just fat around a neuron. It's actually more like if you picture the roll of paper towels in your kitchen, where that central cardboard tube is the axon, and then the spiraling wrapped paper around it, that's actually what myelin really looks like. It's these spiral wraps of membrane.
Oh, okay.
You can add more or subtract more of that. You can also change the length of these sheaths. And all of these sort of physical, geometric changes to myelin will change the underlying conduction velocity. So you can perfectly tune a circuit and how, the speed of all the signals propagate together by changing myelin. So this is actually
Oh,
actively
wow.
happening in our brains.
Yeah, and then do you mind explaining a little bit the methodologies for like how you're like tweaking the myelin, kind of like the experimental part of it?
I said at the beginning, I'm a cell biologist. I care about how
Yeah.
cells change shape, right? And so in my graduate training, we were thinking about cancer cells crawling or immune cells crawling. But now we're thinking about a cell like an oligodendrocyte. So if you want to picture this in your head, an oligodendrocyte that makes myelin, it's almost like an octopus, except it has
Oh, okay.
more than 8 tentacles. It has, dozens of tentacles, whatever you would call that. I don't
So
know
like a
the Greek
sun?
term. Yeah, so each of those individual arms that it's sending out or tentacles that it's sending out interacts with axons, wraps around the axons, and then extends. Because basically a single cell has all of these myelin sheaths that it's connected to, that it is actively supporting. And so that's a complicated cell geometry because those sheaths, like I said, they wrap around the axons too, they grow longitudinally. So thinking about this as a cell biologist, one of the things we try to figure out is what are the cellular processes that cause these cell shape changes to occur, to build myelin or to rearrange myelin. One of the major ways that cells change shape and move is using a cytoskeleton. That's the cellular skeleton. Same is true for immune cells. This is what I studied in grad school. Now applying this to oligodendrocytes. If you think about the cytoskeleton or actin filaments that make up a cytoskeleton, oftentimes you think of muscle where you have cytoskeletal structures, right? Actin
Yeah.
filaments that are generally pretty static. Myosin motors, when you flex, cause the actin to like move sort of laterally relative to each other. That's the way muscles move. But the way cells move when they're crawling around is very, very different. Actually, these actin filaments are incredibly dynamic. They're built like scaffolds that push the membrane of a cell forward. And they're actively being built and they're actively being disassembled as that happens. So in the classic example of a neutrophil chasing a bacteria around, it's building its cytoskeleton in the direction it wants to go. And then when it wants to turn, it disassembles the cytoskeleton in that direction and builds the cytoskeleton in the other direction. I used to play with Lego, so I think a lot about sort of building Lego structures, and then you can break them down into individual blocks. That's the same way the actin cytoskeleton works. And so because the actin cytoskeleton has been studied so much in cells like immune cells and in cancer cells, even in neurons, there are a lot of tools that are existing already for being able to study the role of actin and cytoskeletal dynamics in other cell types. So we've been borrowing some of those tools and we've been creating some of our own tools for being able to understand how oligodendrocytes build and remodel their myelin sheaths using the cytoskeleton. So that's one example.
Where do you see your field going in the next 10 years? Like you mentioned making a lot of your own tools. I'm assuming those are advancing as well because of like, technological changes. Like where do you think students can focus their attention to in the future?
My postdoctoral mentor, Ben Barris, he wrote a really, really beautiful review circa 2008 or something. And it was, glia more than just brain glue. And I always use this quote from the end of it that I put this in my talks oftentimes, but the last line says something along the lines of quite possibly the most important or exciting roles of glia have yet to be discovered. And that was 2008. I think that's still true now, 15 years, 16 years later. There's fundamental things that these cells are doing, oligodendrocytes, astrocytes, microglia, that we don't know about yet. One example, I can tell you this with 100% certainty because we've discovered one of them, which is that neurons are electrically excitable. We've actually found that at least in the peripheral nervous system, sensory neurons don't become excitable on their own. They require secreted signals from the Schwann cells, the myelinating cells that code our peripheral neurons. They require signaling from these Schwann cells to to come online, if you will, during development and become electrically excitable. So this is kind of a mind-blowing thing. We never would have expected this. This is one role of glia that we have figured out by being able to study neurons and glia in isolation away from them. I think there's novel roles. That's one thing. Two is we're just starting to understand the overarching roles of glial dynamics and myelin dynamics in particular in learning and memory. And so by building more and more tools for manipulating myelin, we in the field are going to be able to start testing how important is myelin for different types of learning, for long-term memory, say. I always think about this. I can smell and precisely see the house I lived in until I was 6 years old. All the rooms, I know every single location. Like, how
Wow.
can I do that? Where is that stored in my brain? Is it just these labile synapses, or is there more to it than just that? Could myelin be playing a role in that? And then finally, the third point that I think we all are focusing on more and more is the role of myelin in neurodegenerative diseases. Everyone knows, if you open a textbook, learn about myelin, you know that myelin is lost in multiple sclerosis. This is an autoimmune attack of myelin. This causes severe disability in people living with MS. More and more, we're finding that myelin is affected not just in MS, but in many different diseases of the nervous system, including Alzheimer's disease. And so, we've spent billions of dollars, right, trying to cure Alzheimer's disease by focusing on the amyloid cascade hypothesis, trying to keep neurons happy and alive. Where the field is going now is kind of a more holistic view of the whole brain, not just focusing on neurons, but seeing, can we promote the survival or integrity or resilience of myelin or other glial cell types as a way to reverse or prevent cognitive decline in neurodegenerative disease. I think that's super exciting. There's so much we don't know.
You mentioned that you were just interested by research. Was there something in particular that sparked your interest? And almost how would you advertise research or to a high schooler?
I think part of it was that my dad, up until I was maybe 8 or 9, he was a life science teacher, seventh grade life science teacher, and he would tell us, one of his goals, what he wished he could have done had he not just done that, was to do research and actually be not just teaching the students and inspiring the students, but on top of that, making novel discoveries. So that was probably a bug. I had a little chemistry lab in my basement, but my parents shut it down when they found out that I was lighting fires and I wasn't supposed to be. I think in general, I was just curious and wanted to learn stuff. And then when I realized that what research is not just science that we do through high school and college where you're just learning things. It's actually a creative endeavor by which we try to figure out novel truths of the universe. I mean, that distinction is huge, right? Just learning a bunch of stuff is important. And I'm glad I know a bunch of things because that allows for novel ideas to kind of collide in my brain when I'm sleeping or in the shower or whatever. It's important to know a lot of stuff. You can't just have it on Google. What's different from when I was doing science, in science class, biology class, or whatever. And now what we're trying to do is figure out things, answer questions that we don't have the answer to, make novel discoveries, build new tools. All of that is incredibly creative. As someone who originally was interested in biology and music composition, honestly, I've found both my scientific and my creative outlet here doing this. The side point here is that as a professor, I also get to mentor amazing young graduate students and postdocs and undergrads, and that's just as much fun as making discoveries.
Question is, what kind of skills can high schoolers develop even if they don't have access to a wet lab?
Honestly, less skills as much as just curiosity, passion, wanting to figure out how things work and just thinking it through. That's just, I think, the most important trait in a scientist. Most of the time, what we do it's hard. It doesn't work. I would say probably historically across my whole scientific career so far, maybe 90 or 95% of the experiments that I came up with, they didn't work or they failed to show anything meaningful. But that small 5% of the time that things work, that's where the breakthroughs come from. And so, what got me through that? It was my curiosity and passion. Anyone that I have seen that's had direct personal connections to, family members with Alzheimer's or something like this, that gives you another sort of North Star that can carry you through the dark times of 2-3 years of the PhD or a postdoc. Doesn't make sense. You can't figure this problem out. As long as you have that sort of long-term inspiration and this curiosity that keeps you going, that's a form of resilience that I think is really important in science.
Yeah.
I would just say, read broadly, think about things that interest you. I recently listened to the Walter Isaacson book about Leonardo da Vinci. I recommend
I
you
haven't seen
read it. Yeah,
it.
All his books are wonderful. There's this line in the book where they were going through Leonardo's many notebooks, and there was just a line just out of the blue, describe the tongue of a woodpecker. Just like, he was just thinking about like, well, I wonder what the woodpecker's like, right? And I remember as I listened to it, I was like, huh, that's a really interesting question. I'm curious about that too. As a postscript to the book, Walter Isaacson actually then went and researched it and told us all about the tongue of the woodpecker and how it works. And how, like, it was very fascinating. But I think that's just a nice sort of illustration of how the most successful scientists think. And maybe Leonardo wasn't even very successful. He never published anything,
I
but
know,
he,
it's still
he was successful in
interesting.
other ways. Yeah.
Yeah, I used to get a lot of cold emails from high schoolers or even like undergrads trying to join your lab. What makes someone stand out? Like, how do you pick someone out of the crowd?
The answer to my last question comes through. I mean, that's certainly part of it. This is something that we look for a lot. I would love to get to a position where we have space again to do that because it's great for everyone. High school students are so fresh. It's like the beginner's mind, right? There's no preconceived notions about things. You're receptive, you have interesting ideas that someone who's been in the field and is already kind of indoctrinated with,
A little
neuron
bit jaded.
dogmas. Right. I mean, I think so. I think it's great. It's also wonderful for the students in the lab to mentor. I think for me, the people that we hired, it was really just the emails were personal. Someone had read some of our work and they had questions about it, and that showed curiosity, and it showed it wasn't just someone sending out 100 emails, dear scientist doctor, yada yada yada. I didn't have that opportunity when I was in high school. I didn't live anywhere affluent. Like I said, I grew up in the woods. Things worked out just fine. So I feel like there's maybe a lot of pressure, especially in this area, that students at your stage think that like you absolutely need to have lab experience or otherwise you've fallen off the treadmill already and you can't catch up or fallen off the train, whatever the analogy is. That's just absolutely not true. So don't worry whatsoever. There's going to be so many opportunities over the course of your scientific career. So just keep trying. Hope
Thank you
so.
so much for your time, and I really enjoyed talking to you, and I loved your analogies about science. Have a great rest of your day. Bye.
All right.